I'd like to just say that I'm very happy to be back in Hanoi to see many familiar faces and friends. And I'd especially like to thank Professor Tao, President of HASAR, as well as Professor Sen, Director of the Institute of Clinical Embryology and Histology at VMMU, and also Dr. Chong for inviting me to come back again to see old friends and to enjoy the stimulating discussions. Okay, so as you can see, my title is very different from that in the program, and somehow I think some information was lost in translation. But this is what I'm going to be talking about. How do I work this? Okay. Anyways, so I'll be talking about the focused on one particular topic about pre-implantation testing, because Dr. James Marshall has already given a very comprehensive, wonderful overview of pre-implantation genetic testing of monogenic disease, aneuploidy, as well as structural rearrangements. So I just want to focus now on the structural rearrangements and take you on a historical ride on how this all started. Okay, so now I don't want to go through this because Dr. James has already talked much about it. Basically, you can use blastomeres from day 3 embryos or trophectoderm from blastocyst stage embryos, which are derived by in vitro fertilization or by ICSI. And then you can test for monogenic disorders, for chromosomal abnormalities, and as James has mentioned, you can do them both together now. And the whole idea is to transfer unaffected embryos, whether for single gene disorder or for chromosomal abnormality, and thus avoid affected pregnancies and fetal abnormalities, and avoid having to terminate pregnancies. Okay, so now ICSI, you heard one talk that mentioned that actually ICSI is overused. There's no real clinical utility in many cases, but in the case of pre-implantation testing, there is, because we ensure that the embryos are very clean in the sense that you minimize embedded sperm that may be biopsied and sampled together with your tissue sample. So this is the well, I would say this is a gold standard for PGT. And then the biopsy day 3, and as has been mentioned before, now the gold standard is trophectoderm biopsy. Okay, so now we're focusing only on structural rearrangements in this talk. And the main type of structural rearrangement are the translocations. And there are two types, reciprocal and Robertsonian. And the reciprocal involve exchange of chromosomal material between metacentric and submetacentric chromosomes, whereas the Robertsonian translocations involve the combining of 2 acrocentric chromosomes into 1. And the key thing to note is that there is no gain or loss of functional genes most of the time, and that approximately 4.5% of couples who have recurrent miscarriages actually has one partner or both who is a carrier of one of these types of translocations. And they are very common in the general population, 1 in 500 for reciprocal translocations and 1 in 1,000 for Robertsonian translocations. And so this is just an example to show you a balanced reciprocal translocation between chromosome 7 and 10. And to the untrained eye, they look very, very similar, but to a cytogeneticist who's very experienced, you can tell that actually this part of this is actually belonging to chromosome 10, whereas this part of the chromosome belongs to chromosome 7, and there's a swapping of chromosomal segments of the Q arm of 7 and 10. Now, Translocations aren't the only structural arrangements. In fact, there are many others, and the next most common ones are the inversions. So if there's 2 breaks on one chromosome arm and a swiveling of material between the E and F segments, then you have a paracentric inversion, whereas if you have breaks on different arms and the swiveling of the material between these across the centromere, then you have a pericentric inversion. And depending on whether this material is lost, if it is lost, then it becomes a deletion or a ring chromosome in this case. But we're not going to be talking about these, so we'll just be focusing on the translocations. So now, the ability to test for translocations began with the use of FISH, of fluorescence in situ hybridization. So, and in fact, that was the first method that people were able to determine aneuploidy in embryos. And so basically, whoops, you isolate the blastomere and you fix it, and then you hybridize with a cocktail of fluorescent probes. And in the case of PGT-A or aneuploidy, then the probes can be to the usual chromosomes that you find live aneuploidies, like 13, 16, 18, 21, and 22. And because the blastomere is not in the metaphase stage, what you will see you won't be seeing the chromosomes like I showed you for the karyotype, but you'll be seeing interphase fluorescence signals, and basically you count the fluorescent signals, and up to 12 centromeres have been analyzed from a single blastomere, but over sequential rounds of hybridization. And as James has mentioned, this is extremely tedious, requires extreme technique, and so it wasn't very user-friendly, I would say. Now, for PGT-SR, the probes, instead of centromeres, you will use breakpoint spanning probes. And so, for example, in this case here, you might have a green probe that spans the breakpoint on chromosome 7, and then a red fluorescence probe that spans the breakpoint on chromosome 10. And then if there were a translocation, balanced translocation like this, then in the interface signal you would see green, you would see red, and you would see yellow, because red and green together will give you a yellow fluorescence, right? So, and that allows you to distinguish between carriers and non-carriers. So that's generally the principle. But the problem, of course, is that it's very difficult to obtain probes that are specifically at the regions of your translocations. And so, therefore, this technology had limited application for PGT-SR. So then came along PCR-based methods, which used linked polymorphic markers. And basically, this now enabled one to process the blastomere or the trophectoderm using multiplex PCR method of short tandem repeats. These are polymorphic markers. And then to perform a gene scan analysis and to look at the fluorescent peaks. So here I've just shown an example of a case where the male partner actually has the balanced translocation involving chromosome 12, where the break is on the P arm, and chromosome 22, where the break is on the Q arm. And then basically it involves selection of markers that are present on the P arm of 12, the Q arm of 12, and also on chromosome 22. And then based on the results of the PCR analysis of the embryonic material, then you can see that in the case of the balanced carrier itself, you'll see 2 fluorescent signals for the Q arm of 22, 2 signals for 12Q, and 2 signals for 12P. And this is the normal partner, and these are the alleles. And then if the embryo is unbalanced, what you will see will be that there are actually 3 peaks for whichever segment that is unbalanced. So you'll see that for 22q, there is 1, 2, 3 alleles. And then for chromosome 12q, there's also 1, 2, and 3. So in other words, there's a trisomy 12q and trisomy 22q also. So this was a very simple method. And then for the embryo that is normal or balanced, you would still see only 2 fluorescent peaks for each of these markers. And however, this does not tell you whether the embryo is normal or balanced, right? Now, one of the things that needs to be noted is that although this is an effective method, you need to have the markers that are as close to the translocation breakpoints as possible, because if not, then there's a recombination-mediated misdiagnosis risk, because recombination occurs all the time in meiosis. Okay, and this only allows you to look at the selected chromosomes. It doesn't tell you the state of the other chromosomes that are not involved in translocation. So it wasn't really a very effective method. Then came along microarray-based methods that allowed you to look at all 24 chromosomes and not just the centromeres. So it involved whole genome amplification of the material and the labeling of the material, plus the labeling of a reference DNA with a different color, and the co-hybridization, and followed by scanning on a fluorescent scanner. And basically, you will get results that look like this. And that will tell you not only the state of the translocated chromosomes, whether there's any imbalance of the translocated chromosomes, as seen over here, where there is trisomy 12p and monosomy 22q, but it also shows that there was aneuploidy in the other chromosomes, 5, 10, 13, and 14. Now, but the key thing in this method was to identify embryos that were normal or balanced. And again, this one did its job very well, and not only for the translocated chromosomes, but all the other chromosomes. And again, you were not able to tell between a normal or a balanced embryo at this stage. So, and then obviously now currently the gold standard, as you know, is next-gen sequencing-based 24 chromosome analysis. I'm just showing you one of the methods that are commercially available. And it involves using whole genome amplification again to then generate a library for which you then sequence, and then based on that, you obtain the number of reads. These reads are then mapped back to the genome, and then you basically count how many reads there are for each segment of each chromosome, and then you compare that against a reference, a normal reference, and that will tell you whether your embryo is normal or balanced, or whether there's an imbalance in the translocated chromosomes. In this case, there's a trisomy 1p and a monosomy 4q. And just as in the previous cases, you are not able to tell normal from balanced embryos, right? Because this is only a counting method, so it's quantitative, but it doesn't tell you the translocation state. But recently, this group, and they are not the only group that have come up with strategies to enable us to now discriminate between normal and balanced embryos. But basically, this group utilized a method that uses linked SNP haplotypes to distinguish normal from balanced carrier embryos. And so basically, they go through this similar whole genome amplification and analysis to identify balanced from unbalanced embryos. So that's the first stage. And then after identifying that, in the unbalanced embryos, now they're able to identify the breakpoint much more closely than you would with traditional karyotyping. And so you can identify the breakpoints to within 200 kb, kilobases of the breakpoint. And that now allows you to then identify SNPs that are adjacent to the breakpoints, and based on that, to perform haplotyping on these embryos, and then to identify the healthy embryos and which ones are normal and which ones are balanced. However, now if you look at it, since the embryos will have just the two alleles, the balanced and normal embryos, it's not possible, just looking at the normal and balanced embryos, to identify which haplotype is linked to the normal chromosome, right? So that's why they need to use unbalanced embryos as first to identify the haplotype that is linked to the translocations, and then they will know which haplotypes are not linked to the translocations. So now this is quite complicated, so I don't want to go into detail, but I just want to say that that same group have now identified that among all the possible segregation types from meiosis in a translocation carrier, of course, the two types that will lead to a normal or a balanced embryo, they've called it type 1. And then there are two other types. These are all unbalanced. But they've identified that out of the unbalanced segregants, there are 20 that can be used to identify the haplotype that's linked to the translocated chromosomes. And then they've identified a type 3, which cannot be used. So now, if you are planning to do this, you don't have to worry, because essentially they've worked out which segregants you can use when you want to identify the linked haplotypes. And that's for the reciprocal translocations. For the Robertsonian translocations, it's even easier, because aside for the two normal balanced segregants, there are only 6 other abnormal segregants, and of which 4 can be used to identify the linked haplotype. Now, I just want to use a very, very simple example here of how the unbalanced embryos can enable marker phasing or haplotype phasing. So I've just used the example of Robertsonian translocations, because those are the simplest translocations. And instead of using the SNPs and the haplotypes, which are very confusing, I've just used STRs, or the microsatellites. And I'm just using one marker for each of the chromosomes involved. So, for example, if this is a 13-14 Robertsonian translocation, and you just need to pick one marker here, STR, I just call it STR-A, and this one STR-B. And as I told you, these are the possible segregants. These are the normal balanced segregants, which will result in a normal embryo or a balanced embryo, and these will be the unbalanced embryos. And here is an example of the result for a balanced carrier of a Robertsonian translocation. So you can see that for marker A, for chromosome 13, you have allele 1, allele 2. Marker B, you have B1, B2. Now for the normal spouse, it's A3, A4, B3, B4. This is not important. So long as these alleles are different from these two alleles, that's sufficient, right? So now for the embryos, you will see various different genotypes, but you can see over here that, let's look at this one first. This embryo has already been determined by the quantitative method, whether it's array-based or next-gen sequencing, and by PGT-A, that it is unbalanced. And we know that it's monosomy 13. But when we use these markers to genotype, we can see that chromosome 13 marker, the green one, there's only A3. That's only the maternal, we don't know, this normal parent's allele. This balanced carrier's parents' alleles, none of it is present. Whereas for chromosome 14, now you see this normal parent's allele, and this is the translocated parent's allele. But based on this alone, now you know that this allele is present on the normal chromosome 14 of this balanced carrier parent, because that's the only way it can happen. If it were present on the Robertsonian translocation, then you should see the other allele from this parent. So it's by deduction that you can tell, and therefore once you see an embryo that has 2 alleles for chromosome 13 and 2 alleles for chromosome 14, you know that that embryo is normal or balanced. But because now you have information from the unbalanced embryo, you can tell that this embryo actually is a normal embryo. Now, if you don't understand this, that's okay, because sometimes it takes a little thinking through to understand the very simple logic. So now again, when you use markers, whether they're SNPs or SCRs, there's a risk of recombination-mediated misdiagnosis. So again, your markers need to be very, very close to the translocation breakpoints. The further away it is, the higher the risk of recombination. Now, I just want to move on to show that actually, at the same time, another group published a method that actually distinguishes between normal and balanced carrier embryos by breakpoint junction short read sequencing in PCR. It's a mouthful, but basically they just use sequencing now to identify the breakpoints. But because it involves short read sequencing, it's kind of tedious, because you need to use tricks like mate pair sequencing to then narrow down the fragments that contain the junctions, and then slowly use nested PCR to then identify the breakpoint fragment. You sequence it, then you identify the exact sequence, then after which you can then design PCR primers to amplify the junction fragment. And then again, you need to first do the PGT-A to identify the embryos which are balanced euploid and those which are unbalanced. But now, because you have sequenced the junction breakpoint, you do not need unbalanced embryos anymore to help you to identify and discriminate between normal and balanced carriers. And so after you have these, all you need to do is to perform a separate PCR to identify which ones are the carriers and which ones are the non-carriers. So here's an example. Sorry, I'm just going to carry on with this first. So another group just very recently have come up with a similar method, basically sequencing to identify a breakpoint, but using a long read sequencing and PCR. Now this makes it simpler because when you have long read sequencing, then you can basically sequence, and if you've done sufficient sequencing, you'll be able to identify the breakpoints. So in this case, after you've sequenced and mapped back to the genome, you'll identify exactly where the breakpoints are. And then again, now you need to just design primers that flank the breakpoint. So this primer 1 and 2 flank the breakpoint on the normal chromosome, 3 and 4 on the other normal chromosome. And then, if you pair primer 1 with primer 4 and primer 3 with primer 2, and you obtain a positive PCR fragment, then you know that that embryo actually carries translocations. Whereas if you don't, then you know that that embryo is normal. It doesn't carry any translocation. So this is a much simpler strategy. And again, you need to first identify the unbalance from the normal balance, and then after that, perform the breakpoint PCR. Now, in this case, the authors, just to prove the principle, they performed breakpoint PCR for both the normal balanced embryos as well as the unbalanced embryos. And here's just an example. So E1, embryo 1, chromosome 1 fragment is positive. The derivative 1, the junction PCR, is also positive. Chromosome 18 is positive. The derivative junction 18 is positive. So this one, if we assume is normal or balanced, then this embryo is a carrier of a balanced translocation. Whereas this embryo, on the other hand, embryo 3, is positive for chromosome 1 PCR, derivative 1 is negative, chromosome 18 is positive, derivative 18 is negative. So this embryo 3 actually is a normal embryo. So that's how you can distinguish between them. Okay, so I want to end by just this question. So why is there a need to distinguish normal from balanced translocation carriers? Well, those who say no, they say it because they feel that normal or balanced chromosomes is okay because these are all clinically unaffected embryos. But then I believe that there are those who feel that yes, it's useful to distinguish normal from balanced translocation carriers because not all balanced translocations are benign or harmless. Because even though a carrier may be clinically normal, the translocation may have disrupted a tumor suppressor gene, for example, and they may eventually be at a high risk for some malignancy. So we don't really know that. But if we're able to sequence the breakpoints, then we'll be able to know for sure whether the translocations actually disrupted any functional and important genes. Secondly, carriers of balanced translocations, if they are these embryos, they will grow up and experience exactly the same reproductive frustrations of their parents. So that's another reason why it would be good to be able to distinguish normal from balanced translocation carriers. And finally, X-autosome translocation carriers can manifest clinical phenotypes that are associated with aberrant X inactivation. So whatever segment, autosomal segment that is present on the derivative X chromosome could be inactivated, the genes on there, and those X chromosome segments that are translocated onto an autosome may not be inactivated when they're supposed to be. So those might create some clinical issues with them too. So in terms of future developments, and this is probably my last slide, I see that as it is already, things are moving towards single technology platforms for both for PGT-M, A, and SR, where you can see that you can have combined PGT-A and SR with the ability to detect and discriminate between balance and carriers. Or you can have combined PGT-A and M with ability to discriminate between normal and affected embryos for single gene disorders. But I see that eventually there will be more people embracing long-read sequencing simply because it's simpler to identify translocation junction sequences. And the other thing that's good about long-read sequencing is that it also eliminates SNP haplotype phasing for those who want to use haplotype-based methods of discriminating between normal and balanced embryos. Because when you read the entire strand, you actually look at all the alleles of the SNPs that are adjacent to your translocation breakpoint. And then finally, now because methods like the nanopore, they've come up with strategies to enable targeted sequencing of mutation sites and the translocation breakpoints. Whereas if you use the current next-gen sequencing for Illumina as well as Thermo, you have to sequence the entire genome, and you have to be able to have deep sequencing in order to be able to identify the genotypes of each of the SNPs that you want to use. And so that's a lot of sequencing firepower. However, with new algorithms designed to enable just targeted sequencing of the sequence regions that you want, then that will lead to reduced sequencing, higher read depth, and faster turnaround time. And you only see what you want to see, and you don't care about the rest of the genome. Okay, so I think I will end there, and thank you for your attention.